Okay, welcome everybody to today's Neuromodulation New York City 2020 session on navigated transcranial magnetic stimulation in the clinic and the lab. We'll provide an overview of current practice and future directions. Welcome everybody to our Zoom session. We're really excited to have you all here and present you with latest insights in our field. I am Bas Neggers, your session chair for this afternoon. I hold an associate professorship at UMC Utrecht, and I'm also founder and CTO of Brain Science Tools BV. The session is also supported by Soterix Medical in New York City and Neurosoft from Russia. For our first message to all of you, you can ask questions in between sessions through chat. We will collect the questions and handle them after the presentation one by one. Our co-host Alexei, a Zoom expert, will collect them, and we hope we can answer all of them. Yeah, this is for us the first time, so we hope this goes well. As a first speaker, I'm proud to introduce Nikolai Smirnov, one of the first people from the first hours of Neurosoft and an expert in TMS and EMG. And he will introduce you into TMS protocols and some first impressions of what's going on in the brain during TMS. Nikolai, the floor is yours.
Thank you, Bas. And hello, everybody. I will share my screen. So, Bas, Alexei, you should switch off. Yeah, right. Thank you. All right. And I will share my screen. Okay. All right. Now, all right. So my name is Nikolai Smirnov, and I'm from Neurosoft, and I was involved in the development of several generations of TMS systems since 1996. And I will speak today about what happens in the brain when we deliver TMS pulse. We will look today inside of TMS machine. We will look at different effects of transcranial magnetic stimulation. We will look at different TMS protocols. So I will do kind of short overview about the TMS. And I will start with this video. And in this video, you will see a very simple way how to explain what is TMS.
This is transcranial magnetic stimulator. This is TMS coil. When I switch it on, it gets ready to deliver the stimulus. When I press this button, it delivers the stimulus. When I press this, it also delivers the stimulus. Here I can change intensity of stimulation. Here is the bulb. This is just the same bulb you use in your car. Here we have several wings of copper. So what I do, I just put it on the coil and deliver the stimulus. You see, the bulb is blinking. And I even don't need to touch it. Even there is some distance, some gap. It is still blinking.
All right. And what actually TMS machine is? It is a very big box. And if you open this box, you will see this big capacitor inside. This gray cylindric stuff at the left is a capacitor, and the rest of space is a charger. So basically, what you have a charger which charges the capacitor, and then in the very middle, this white piece is a key, is a thyristor, using which you can close the circuit. And then the energy will go from capacitor to the coil, and the coil will deliver this energy to the space, and this energy will be enough, for example, to switch on the bulb. But what I will do next, I will stimulate myself here.
Now I will stimulate myself. First, I will stimulate here. Look at my fingers. You see? When I deliver the stimulus, electromagnetic field goes through my clothes, muscles, reaches peripheral nerve. Nerve gets excited. Excitation wave goes through the nerve, reaches the muscle, and muscle is constructed. I can do the same when I'm stimulating my motor area. So if I'm stimulating here, where is my motor area responsible for my hand is muscles are constructing and hand is moving. If I'm stimulating here, nothing happens. If I'm stimulating here, nothing. Only when I'm exactly on the hot spot.
Right. So, and here is a very classical presentation of what's going on. I will just briefly repeat this. So this yellow arrow shows direction of electrical current. It's really brief. It is just 380 microseconds duration of the pulse. But the voltage on the capacitor is very high, is about 2000 volts. So what you are doing, you are delivering this high voltage to this coil. These coils create very intensive electromagnetic field, and this electromagnetic field can easily go through the skin, bones, and reach conductive tissues. In this case, it could be cortex, and it induces electrical current inside the cortex, and it's kind of switching off on the bulbs inside of cortex, and depending on where your coil is, you can switch these or these neurons. You can switch on these or these bulbs. And now what I will do, I will put some electrodes on the patient.
This is TMS machine.
And I will record MEP.
This is a TMS coil connected to the machine, and this is an EMG amplifier, which is connected to this PC. TMS is also connected to this PC. On the PC I have the software which can talk to the TMS and to the EMG. And when I deliver the stimulus, EMG machine can pick the signal and show it on the screen. I will put electrodes on the volunteer. So this is the ground. This is a minus on the muscle and plus on the tendon. I take the coil. The intensity is 50 now, and I put the coil on this spot. This is the motor area of left hemisphere responsible for the right hand movement. And I deliver the stimulus. So what I see on the screen is called MEP.
Right, this MEP. This is kind of wave. The flat part at the beginning is just a rest EMG, which reflects zero activity in the muscle, and then you see the wave itself, MEP, motor evoked potential itself. So it has a latency. It's a time from the stimulus delivery to the wave, and then it has amplitude. So you can measure these two parameters, and you can have numeric values for this MEP, latency and amplitude. So message number one of my speech is TMS really induces electrical current in the cortex. Let's try to understand what's going on. So when I deliver TMS pulse, somehow I get upper motor neurons excited, and then excitation wave goes down via brain stem and reaches peripheral motor neuron. Then peripheral motor neurons get excited, and then excitation wave goes down via peripheral nerve, reaches the muscle, and then I have two electrodes on the muscle, and I have EMG amplifier, and I can record MEP response. right? And now what we will do, we will try to modulate this response with several tricks.
Now we will try to modulate this signal, this response. So please construct your muscle, just do like this. Okay, thanks. As you can see, the amplitude of second response is much bigger than first one. The first was 1.8 millivolts. The second is 6 millivolts, 3 times bigger.
So when the muscle is constricted, you clearly see that amplitude of response is bigger, because when the muscle is constricted, neurons are pre-activated and you deliver the pulse on the active brain. So let's see another experiment.
Again, this is the response. And now we will try to facilitate it mentally. Now you don't construct the muscle, but you think that you are constructing the muscle. You should see how it's moved.
Well, it's kind of imaginary construction. So she is not constricting the muscle. You see the hand is relaxed, but mentally she is thinking about constriction.
Clearly see how your finger is moved. More. You see, this response is also bigger than baseline. The first response is non-facilitated. The second is physically facilitated, and the third is mentally facilitated. The first is 1.8, the second is 6.3, and third is 2.35.
So as you can see, MEP amplitude really reflects overall activity in the stimulated area of the cortex. So then bigger the response, then more active the cortex under the coil. But now we will do something which is completely opposite.
Another test. First I will show you EMG. This is the EMG. This is what we pick from the muscle. Relax, please. Fully relax. Okay. So this is the EMG, the baseline. So if I ask you to construct the muscle, you see this is the EMG. Relax. Nothing. Construct. EMG. Okay. Relax. Now I will deliver the stimulus. This is MEP. And now please construct the muscle, and I will deliver the stimulus during voluntary construction. Okay, and save it.
So let's look at it. So you see activity before the response there is activity. She was constricting the muscle. She was really constricting the muscle, and there is the EMG activity. Then you see response itself. It looks slightly different because here I use different scale. But then after the response, you see very flat piece of 200 milliseconds. She was constricting the muscle. She was voluntarily constricting the muscle. She was thinking she's doing that. But in reality, the muscle had no activity. The muscle was relaxed. So I switched off with TMS pulse. I switched off activity in the muscle for a very long time, for 200 milliseconds, as you can see on this scale on the screen. And if you would do the same using different intensity of TMS pulse, here there is responses with 3 different intensities: 70%, 75%, and 80%. You see that cortical silent period is getting longer. We call this silent period because it's period of silence after TMS pulse. This is inhibition. This is a way how we can see at the TMS pulse. So the message number 3 of my talk is very important. TMS always evokes both excitatory and inhibitory processes in the brain. So each time when you deliver TMS pulse, visually, normally you see only excitation. Normally you would see only movement of the muscle. You think always when you do TMS, you always think about excitation, but in reality you have two processes, excitation and inhibition. And I can say that excitation is even longer than inhibition is even longer than excitation. Now let's speak about repetitive TMS, because before we were speaking about single pulse TMS, but before we speak about repetitive TMS, I must tell you about TMS stimulation patterns.
Stimulation modes. Now the machine is in single pulse mode. So when I deliver the stimulus, it delivers me one pulse. One press, one pulse. When I push this button, it goes to repetitive mode. Now it is 10 pulses per second. And if I press the button, it delivers continuous repetitive or continuous rhythmical stimulation, 10 pulses per second. When I press the button, it starts stimulation. The next mode is stimulation by trains. So here I set the frequency, which is 10. I can change it. For example, now I will do 9. Then I can set train time. For example, 3 seconds. Then I can set pause time. So, and then I can start stimulation. Now it will stimulate 9 pulses per second. It will make 3 seconds train, and then 2 seconds pause, like this. 3 seconds train, 2 seconds pause, 3 seconds train, 2 seconds pause, and so on and so forth. And final is theta burst mode. Instead of 1 pulse, there is burst of 3 pulses.
So, and now, let's think about what's going on when you do repetitive stimulation. So with single pulse stimulation, you can evoke in the brain the process, excitation process or inhibition process. You can increase or decrease activity. What happens if you would deliver several hundreds or several thousand pulses to the brain? And here is a paradigm of very typical RTMS experiment. So what you can do normally, you can measure baseline MEP amplitude. We remember that MEP amplitude reflects activity of the brain. Then higher amplitudes and higher activity in the brain. So you measure the baseline amplitude, and then you deliver, let's say, 1,000 pulses of 10 pulses per second frequency at certain intensity. And then you can measure the MEP amplitude every single minute after this sequence of 1,000 pulses. And in this case, you would measure kind of increase of activity in the brain or decrease of activity in the brain after this neuromodulation session. And in this particular case, you can see that all responses after RTMS were higher by amplitude than the baseline MEP. So you can say that this particular neuromodulation experiment showed that MEP increased, activity in the brain increased after RTMS. And there are hundreds and thousands researchers, publications, articles showing how RTMS modulates activity in the brain. If you go to PubMed and if you type TMS LF, TMS excitation, TMS motor, you will always find hundreds of articles like this one. For example, this one showing how high frequency TMS increased activity in the brain. So in this case, 8 healthy volunteers received either 150 or 1,800 stimuli of 5 Hz in 2 separate days. And as you can see, that long session of 1,800 stimuli of 5 pulses per second induced increase in MEP amplitude, and the effect was stable for at least 30 minutes. This is another one showing how low frequency 1 pulse per second works at the brain, and you see that 1 pulse per second over the left premotor cortex suppressed amplitude of MEP, and effect outlasted for at least 15 minutes. So in some situations, TMS increased activity in the brains. In some situations, TMS decreased activity in the brain, and it is not very well understood why it's going on. But you can think this way. Let's say you have 1 pulse per second stimulation. In this case, about 20 to 30 milliseconds after one stimulus, the brain stays active, and then about 150 milliseconds, brain stays deactivated. And if you would do this repeatedly once in a second, you will see something like this one. So most of the time, the brain is low state, is inhibited. But when you do high frequency stimulation, then each stimulus activation period of stimulus overlaps inhibition process, and brain stays overactive most of the time of such stimulation. So, and it is postulated in many articles in the literature that high frequency stimulation in general has excitatory effect on the brain, and low frequency stimulation in general has inhibitory effect. So if you would go to any RTMS article or in any RTMS book, you will see these 2 letters, LF or HF, which stands for low frequency or inhibitory TMS, or high frequency or excitatory TMS. And very important milestone of TMS was early 2000, 2007, 2006, when TMS, when researchers started to apply TMS for depression. And this is the result of one very important trial of, in this trial, about 300 patients get RTMS, and they get RTMS for very long time, for 6 weeks, every day, which is 30 sessions, and they get every day 37 minutes of 10 pulses per second stimulation. And this was in really depressed and major depressed population. And as you can see on this slide, people, the patients who get active TMS significantly benefit from RTMS if you compare with the sham group. About 50% of patients get improvement after RTMS, and the improvement response and the improvement rate was also about 50%. So it was after this publication, RTMS was cleared for depression treatment in USA, and after this publication, TMS get very popular because from this point on, we can use RTMS in daily psychiatric practice. So message number 5 is that says RTMS is well-established tool to treat depression, and in depression, dorsal left prefrontal cortex is the target. And the question is, why is that? But before we answer this question, let's look at another way to measure activity in the brain. After RTMS, you can measure EEG. So in this particular experiment, you see TMS machine, you see TMS coil, which is above the motor cortex of this patient, and you see EEG machine, and in this case you can record EEG during the TMS. And here is a screen of EEG, and on this screen you see EEG traces, and here is the average traces. And by this red cross marked the place of stimulation, and you see at the place of stimulation the average EEG response is highest by amplitude. But also you can mention that around the brain, literally everywhere in the brain, you see responses. So you are stimulating one area of the brain with TMS coil, but we have responses in other areas of the brain. So, and this brings us to discussion about networks. So we all know that the brain now can be presented as a network which has different nodes and connection. And when you stimulate one node of network, you would definitely have effect on another nodes of the network. And this is exactly what's going on in depression, because we know that anterior cingulate is the area, subgenial cingulate is the area responsible for the mood and for depression, but we cannot reach TS with TMS this area. So what you do, you stimulate DLPFC, which is very very close surface. It is much more frontal, and you stimulate subgenial cingulate as a next node of this big network. Same thing happens when you do RTMS in stroke. In stroke, we know that motor areas of left and right hemisphere are very well connected, and this is negative coupling. When one hemisphere is active, another is deactivated, and opposite. So when there is stroke, all hemisphere is always deactivated, and what you can do, you can decrease activity of healthy hemisphere, and by doing that, you would increase activity in affected hemisphere, and it will have therapeutic effect. So here, again, we use network approach. So message number 6 is that stimulation of one area of the brain leads to changes in other areas of the brain.
Nikolai?
Yeah.
There are a few more minutes for questions, so maybe finish this slide and then we'll take questions.
Right. Yeah. I will skip a couple of slides, but what I will do, I will go to TBS, theta burst stimulation, which is important part of this. So it's just a couple of minutes. So theta burst stimulation, as I showed you, is when you replace one pulse in classical RTMS by 3 pulses. So it delivers very brief sequences of the pulse. And TBS has the same effect as a classical TMS, but it is way shorter. For example, in this case, ITBS has intermittent TBS has excitatory effect on the brain, as you can see on this slide, but it lasts only 3 minutes. And CTBS, it is just 40 seconds, that it has inhibitory effect. And as you can see at this slide, this effect also lasts very long after theta burst stimulation. And we have very nice research, which is just published in 2018 recently, where they tried to apply these TBS, theta burst stimulation, in depression patients. And as you can see here, on more than 400 patients, the result of classical 10 pulses per second stimulation was exactly the same as a result of TBS. And the message number 10 here is TBS, theta burst stimulation, is significantly faster than conventional RTMS, just 3 minutes versus 19 or 37 minutes. But it has exactly the same behavioral and net effect. So this is take-home messages. High frequency stimulation increases activity in the brain. Low frequency stimulation decreases activity in the brain. You can stimulate one node, you can stimulate one area of the brain and have effect on other areas of the brain. To achieve neuromodulation effect, you should deliver to the brain significant amount of pulses, several pulses, do neuromodulation sessions, long time days, or even several. That was all what I wanted to show you today, and I want to invite you all to our courses, which we have every year, and this year it's planned for beginning of December 8 and 9. And I hope that we will be able to run these courses physically, not only online as we do it today. And I heartily invite you, everybody, to very nice city, Utrecht, in Netherlands, where we have these courses. Thank you very much. And now I'm ready to take the questions, please.
Thank you, Nikolai. We have one question from Akhil Mohan. And his question is, what does the post-activation potential after the silent period signify physiologically? Can you allude on that?
I'm not really getting what doesn't mean. So, but if we look again at the silent period response, so we just see how EMG is going back. So at the beginning of the response, we see EMG, then we see MEP, then we see flatness, and then again EMG, because the volunteer, the patient, is still constricting the muscle. What we see it is just EMG.
All right, so it's restoration of the normal physiological EMG.
Exactly.
From the patient. All right, then I would like to continue with Bas Neggers' talk. Bas Neggers is, thanks again, Nikolai. Bas Neggers is CEO of Brain Science Tools, a company that produces neuronavigation and distributes electrostimulation equipment in the Netherlands. But he is also associate professor at the University Medical Center in Utrecht. And he will be talking about navigated transcranial magnetic stimulation in the clinic and in the lab. I would like to give the word to Bas.
Thank you, Jort, for this great introduction that I couldn't do myself, of course, as the session chair. I hope everybody can hear me. All right. Indeed, I'll continue to show you how you can use magnetic resonance imaging to augment TMS, both for clinical and research applications. A little bit about myself. As said, I'm associate professor, biophysicist who became neuroscientist. I specialize in neurostimulation, and I also bring applications from the clinic to the market. So we also have a spin-off company that's doing that. So my talk will also show a bit what we do at the company. It's a bit of a mix. All right, and the UMC Utrecht is a large academic medical center in the heart of the Netherlands. So using MRI for navigated TMS applications, how can we accomplish that? So first I want to make a distinction between offline and online applications of MRI and TMS. With offline application, we mean to say that we first acquire MRI images of different modalities, structural MRI, functional MRI, and then use that information to guide a TMS coil to a certain location or orientation or what's more. So we use MRI to inform our TMS experiments or treatment. Online MRI TMS is something different. There you actually apply TMS inside an MRI scanner, so at the same time. There you can see what's really going on in the brain when you give a pulse. So I'll give some first results on that too. So that's online MRI TMS. So those things use MRI in a different way, before a TMS experiment or during. So I hope that's clear. So let's first look at offline MRI guided TMS. What it is, why we need it, how it works, and what the added value is. So first, we've seen in Nikolai's great talk what TMS can do, how it evokes activity in the muscles, etc. Then you can search for a spot on the scalp overlying the motor cortex and see what happens. But often that's not the case. So often you stimulate another brain area related to vision, to attention, to memory, or mood, or anything else. And there it's really hard to know where to place your coil. So how do people do that in general? So the classic motor hotspot method is that you first look for a spot that moves your fingers, and then from there you go either 5 centimeters laterally or anterior from that, or another direction, and hope that you are roughly in the right vicinity. That's a really crude method, but still quite popular. The 1020 EEG system, you can use an EEG cap, place it on the head, and then pick one of the coordinates. This roughly corrects for head size, but that's about it. It doesn't take into account the morphology of the brain. Some people have been using fiducials that you stick to the head and then an MRI scanner, and then move with respect to that to roughly come to a region that you want to stimulate. But at the moment, the best practice is really that you acquire an MRI scan, measure the head of the position of your patient in the lab or the treatment setup, also measure in real time the position of the coil and visualize it on the screen. So this is called neuronavigation or MRI guidance. So that's the best practice at the moment. So these roughly 4 ways you have to determine where to place your coil. So what is MRI guided TMS exactly? So let's have a closer look at that. So you can use an individual MRI or functional MRI scan to guide a TMS coil to its target in the brain. As I said, so it has a really, it's really precise in targeting in the intended area. It's a few millimeters precision, so that's the best we can do. And you can see in real time what you're doing with respect to a brain area. And you can also use functional MRI or DTI data on top of that to guide the placement of your coil. Of course, we have a system like that. It has been developed and researched long ago, and that we've now, with our spin-off company, brought to market. It's installed in more than 70 hospitals worldwide. And it closely integrates with Neurosoft's TMS and especially EMG. So more about that in the demo workshop at the end of our session. Why do we want this? It's a bit of effort. You need to make a scan of about 5 minutes at least. So it's some more work, some more money, but what do you gain? Well, seemingly the problem of placing a TMS coil is simple when you have an MRI scan. You have a brain region of your patient, a coil, so you simply put it on there. Well, it's not that simple. Even when you have an MRI, where is the position on the scalp overlying this area in the brain? So you need to co-register the space your patient is in with the space he was in the MRI scanner. So that's the main job that MRI guided TMS has to solve. And conceptually, you need it because no brains are alike. So at the right-hand side, you see 2 renderings of the cortical surface. One is mine, so 2 subjects inflated to the same size. And you can see that that the central sulcus, for example, here, can already be in a very different location on the AP axis from one person to the next, even when their heads are of the same size. Not to speak of the frontal cortex that's has a really personal gyrification. So you really need to take into account individual morphology to place your coil correctly. So how does this work really? So you basically digitize a set of points in the MRI scan that's visualized on the screen over here, and you measure the same positions on the head with a digitizer, a 3D digitizer. Once you've done that, in the navigation software, you can visualize any tool with respect to the head. So this is also used in neurosurgery, for example, MRI-guided neurosurgery. In practice, using our system, this is what you do. You make a short scan, a few days or weeks or even a year before the TMS session is fine. You then prepare the data, place these markers, measure them when the patient comes in. The software computes how to relate these point clouds to each other. And then in real time, you can guide the TMS coil to its goal, say the dorsolateral prefrontal cortex when you're treating depression. So that's roughly the workflow. It's relatively fast. So this preparation takes, when you practice, about 5 minutes in our system. So some practical notes again on MRI. So when you make an MRI for neuronavigation, as neurosurgeons also know very well, you need to make sure your resolution is high enough, about 1 millimeter between slices. So the quick neurological exam that's often made with 5 or more millimeters in between slices is not good enough. So there you can spot, for example, brain damage or a tumor quickly, but it's not enough to reconstruct the skin or the brain surface. So you need a better resolution and cover the nose and the ears as well. So that's the difference. But this is a really similar practice as for neurosurgery. So what options do you then have when you want to choose a target? You still have to know your neuroanatomy. So you can take a coordinate from literature. Our software supports that too. That is then warped to the individual MRI scan of your patient, which basically places a group estimate from literature on your patient's brain as accurately as possible. So that's supported by our system, among which the most popular treatment targets. This is fast because you only need a 5-minute T1 weighted MRI, and therefore a relatively inexpensive solution. You can look at the cortical morphology of your patient, really study your neuroanatomy well, and then individually place a target based on what you know about where the major gyri and sulci are. That's also a very good method, fast and economic. The most precise, as has been shown, is functional MRI, when you can do an individual fMRI activation task in an MRI scanner, for example, for memory or attention or any task you want to investigate, and then you can localize individual functional brain subdivisions very accurately. This is called fMRI guided TMS. Our software supports that as well. So here you see a paper by Alexander Sack, where he has been comparing how many subjects you need to get an effect of TMS on visual attention significant. And he found that when you use the EEG cap method, you need about 48, I think, subjects to get this effect significant, this behavioral effect, with Telerec coordinates projected to your individual patient's brain. It's only 13. With MRI anatomical, it's about 8, and he claims you only need 5 subjects to get this effect significant when you use functional MRI guided TMS. So there's clear evidence that that's the best method to go. Of course, you need to pay a bit more money. You need to do a little more effort to do fMRI before your treatment session, but it pays off, and at least in terms of accuracy. Now on to the clinical use of MRI-guided TMS. In clinical application of TMS, there's also a lot of benefit to use MRI guidance. I'll show you a couple of cases from depression treatment, psychosis treatment, tinnitus treatment, and one other case. I can skip the slide. So as Nikolai already has shown you, that O'Riordan and many others by now have shown that RTMS treatment of depression on the dorsolateral prefrontal cortex after several weeks gives a marked reduction in symptoms of depression. And still the coil is often placed using the 5-centimeter rule, so the spot 5 centimeter anterior from where you evoke thumb movements and then stimulate there. So that's still most often used. However, it's now quite clear when you have a look. Here in blue, these dots are locations that were established using the 5 centimeter rule, and the red one with neuronavigation is where you actually want to be. So they use neuronavigation to follow where you end up with the 5 centimeter rule. And you can see it can be really inaccurate. So when you stimulate here, you don't do much at the LPFC. And here you see the same data in 3 different views. There are 3 more papers like this that show how inaccurate it can be. So also, not only in terms of accuracy, but there's even a 3rd paper now where they compared MRI-guided TMS with blind TMS, especially the paper by Paul Fitzgerald from 2009, where he showed that MRI-guided TMS shows up to 30% more symptom reduction at week 4 when you use MRI to guide your coil. So that's convincing. And there are 3 more papers by now, so this is even outdated, the 2, that show something similar. So that's also showing that the clinical efficacy of your TMS treatment will benefit from a short MRI scan up front. I'll skip this for time's sake. The TMS treatment of psychosis that we have also spent a lot of time in some of the biggest clinical trial can also be aided by anatomical MRI or functional MRI. A meta-analysis of all clinics up to 2012 that use TMS treatment of psychosis have shown that there is at least a net effect from those papers. However, our own trials were not too successful, so we have a bit of doubt here. And we used a coil placement based on Broca or the temporoparietal junction to guide a TMS coil. However, when you compare guided with unguided TMS, there's still better results in guided studies. So that's something to take home. But in general, on psychosis treatment of TMS, we ourselves are not that positive anymore. I have to be honest there. Another case is tinnitus, where you stimulate the primary auditory cortex of patients suffering from ear ringing. There we did see efficacy of this treatment. It was better than placebo treatment for only the heavy tinnitus sufferers, but it was not much. It was 5 to 10% at most. So scientifically, it's interesting to further improve this. However, whether this is clinically viable and useful, that's still open for debate. But here also we used MRI guidance to stimulate Hassel's gyrus, where the primary auditory cortex is located. So here you can also use MRI to improve accuracy of your coil placement. Yeah, so this is the data where you see that for patients that had severe problems with tinnitus, we saw a small effect that was only significant there. One of the largest trials to date. With Parkinson's treatment, people either target M1 or the supplementary motor area that you can also easily find by following the ventral tegmental line, going up to the cortex and place a target right there. Or you can use functional MRI with a stop task to individually localize it and treat for Parkinson's. How well that helps is not that well understood yet, but at least you can use MRI guidance to get to your goal. So these were 4 clinical cases where MRI guidance seems to be useful and is actually used increasingly. Yeah, so I'll take a breath and move on to other clinical applications of MRI-guided TMS, more diagnostic. Let's have a look at neuronavigated motor evoked potential mapping. So you have seen Nikolai's great talk that when stimulating the motor cortex, you can evoke a signal in the corticospinal tract that goes to one of your muscles and evokes a signal looking like this, the motor evoked potential. You can do this during neuronavigation, so during tracking of the coil and the head of your patient. Our system can do that too, and you can then compute the center of the induced electrical field and mark that on the cortex with a little sphere right here. Here we did this for one of the thumb muscles, and you can indeed see that M1, this is known, where the motor hotspot, where the thumb is located, is marked. So you can individually map, functionally map, non-invasively this region. That's a pretty cool trick. That's not only cool, you can also use this for quite a few things. So you can follow recovery during stroke rehabilitation, and you can plan surgical interventions with it. Let me go into this a bit more. So when you plan glioblastoma resection or tumor removal, a life-saving intervention that neurosurgeons do, it's important to determine which tissue is still intact and which tissue must be spared. So this is usually done classically with an electrode and an open skull. The patient is awake when you do this, so this is not very pleasant. And then they see when a limb or a muscle moves when you stimulate. So this is risky and expensive. You can actually do this these days with navigated motor-evoked potentials, and maybe in the future even prevent the awake phase of the surgical intervention. So that's an application we're working on closely with neurosurgery here in Utrecht. Another application that's newer is that you can actually follow what happens in the brain of people with brain damage, for example, after a cerebral vascular accident or a stroke. So you can follow in a subacute phase to the chronic phase in a rehabilitation setting, so not even you don't even need an MRI scan anymore after the first one, and follow over time how these muscle representations reorganize as the result of rehabilitation schemes. So Jort Vink, after me, will tell a lot more about his trial he's doing with his equipment, and I won't take away his findings and leave that to him. But this is also a really cool new application of diagnostic MRI-guided TMS. How does this look? So this is the solution we now CE certified for medical use in the EU last year, and a few weeks ago we were really happy to announce that this solution was also FDA cleared for clinical use in the United States. So that's and also for depression navigation use. So this, we're really happy with that. So it's also cleared for clinical use in Brazil and Australia as well. So our customers from and partners from down under, it's available to you as well in this form and shape. So here you see the setup with a monophasic TMS device, our navigation system, the software, the NeuroMap software by Neurosoft, and the 2-channel EMG system. So this you can see more on our website. So we really strive to bring improvements from the lab to the market as well, and we fund new research with this. So the future, where is this field going? So I've showed you the past, the present, and now let's go to the future. That's already here in parts. So you can now determine with MRI where to put your coil. You can map muscle areas, but what's the dose? So how much intensity do you want to put in someone's brain? The motor threshold is really crude, everybody knows that, not that reliable. So many groups, there was a session yesterday by Axel Thielscher on this as well, now try to compute with a lot of physics what currents a certain coil induce in a volume, in a conductive volume like the head is. You can do that. We are doing that as well. So you use the principle of Faraday to induce currents in the brain, and you can also compute how big they are. However, the brain is not homogeneous, so you can also take into account the individual morphology and conductive properties of someone's head tissues, and then compute in the skull, cerebral spinal fluid, and the gray matter of the brain, the cortical surface, how big the currents are that you actually deploy. So this is the term in this equation that's due to the coil itself, and this is the resistance that causes additional current accumulation in the brain and distorts the field a bit. So these two parts you can model. First you compute the field induced by a figure-of-8 coil, then you mesh the brain of your patient using his individual MRI into tissue types, and then you compute of these two terms together what the currents actually are. When you want to read more about this, you can read our overview paper in Progress in Brain Research in 2015. So this is a future way, not even that much future anymore. It's approaching the stage where it's built into navigators, where you can determine the dose in in volts per meter in your brain by physics and not just looking at thumb movements. So this is, I think, really where this field is going, to have controlled dose in the clinical setting. So this is also there and coming, so coming fast. So the holy grail for me is that we make an MRI scan and we have a unit that can compute milliamps in the brain, and then you adjust the stimulator such that you get a certain current in the brain where you want it. That's where this field is going, I'm pretty sure. And last but not least, a very cool application we've recently been publishing a couple of papers about is online MRI TMS, where you can apply TMS inside an MRI scanner. So here you see the cover of Neuroimage from a recent paper also by Jort Vink. He will not tell himself after me, but still it's really nice work where we so we know now that DLPFC is an effective target for depression treatment. It's also known that the subgeneral prefrontal cortex in the middle of the brain below the frontal part of the corpus callosum shows abnormalities in depressed patients. It's also known to be involved in the mood network, and it's known to be an effective target for implanted electrode stimulation in depressed patients. It's a bit unpleasant though to open your brain and do that. So, but this offers a few clues why depression treatment might work, but a lot is still unknown of how this works when you apply TMS. So therefore, we've been following the question, what happens when you stimulate the DLPFC in the brain? People have been using this for 20 years now, but no one really knows what's going on. And thus, is there a link with the subgeneral cingulate that's also implicated in mood disorders? So there has been some resting state indirect clues that this is indeed areas that co-vary and are working together, but how is not known at all. So therefore, we built a setup that can do TMS inside an MRI scanner, and we measured fMRI during single pulses of TMS. So you have to take care of a few things. So you have to stimulate in between reading slices, a small dead period. So it's really interleaved TMS. And there are strong forces on the coil that you need to counteract by choosing the coil angle smart. I won't go into that. It's all on the papers we have written. But so don't try this at home without precautions. It can be unsafe when you don't take proper precautions. So how does this look? So here you see an animation also made by Jort with MRI, single MRI coils next to the head, not a birdcage coils, to generate space, and the TMS, an MR compatible TMS coil. We place it at DLPFC right here, marked with neuronavigation beforehand. So we know for sure that it's there. And we also follow the coil in the scanner with a quick T2 weighted MRI and fluid-filled tubes over here. So we know pretty well where we are. So this is then where we would induce currents in the brain for this patient inside the scanner. And we measure fMRI. So over here, you can see that indeed we are able to activate not only DLPFC, but also the subgeneral cingulate region in about half of the 9 subjects we measured here. It was a small study. So it can happen, but not for everyone. So there's variability. But this is the first paper showing that indeed you can induce activation in both regions. So that's important. There's a link.
There's only a few minutes left, Bas.
Yeah, I'm almost done. Thank you for the reminder. So that's cool. We hope to follow this up with a larger study. So the conclusion is concurrent TMS can be used to visualize network activity in response to TMS. TMS-induced activation propagates to the sublingual region, but there is a substantial variability. Maybe this could predict for who TMS treatment on DLPFC could work or not, but that we don't know yet. So this was my presentation. I can take questions.
Thank you, Bas. Very clear talk. There is one question by Felipe. fMRI-guided neuronavigation can inform you where to stimulate the patient, but whenever the coil is in place, how do you determine the coil's orientation?
Very good. So we measure the TMS coil and the subject's head with so-called 6D tracking. So at every moment in time, 100 times a second, we know X, Y, Z of head and coil, but also the 3 cardinal angles, yaw, pitch, and roll. And that's how we reconstruct everything in 3D space. Our navigation system also computes the coil angle with respect to the mid-sagittal plane in degrees, and that's displayed in real time. You'll actually see that during the demo right after Jort's talk. So yeah, that we can also compute. You can also use that to optimize the coil angle with respect to a sulcus. I hope that answers your question.
There's another question by Joel. With simultaneous TMS fMRI setup that you presented, is it possible to target regions which are located more posteriorly, for example, temporal or parietal regions?
Yes, we actually did that for a few subjects in the paper by Antoine de Weyer 5 years ago. Because we have a very flexible setup. These coils are also called flex coils, so we don't use the bird cage. So we can rotate the patient's head like this and then put the coil also on parietal and temporal regions. That's possible, yeah, but it's a bit uncomfortable, so the subjects cannot do that for that long, 10-15 minutes max. But it's possible with our setup, indeed. One of the few setups this works with.
All right, thank you, Bas. There's one more question. Let me find it. Do you have experience with TMS for stimulation of the supplementary motor area in patients with brain tumors? And there is a mention about pre-surgical examination.
I do know that there's an interest by neurosurgeons in this region because you often can have glioblastomas near that area. It's not part of the classical eloquent areas. I don't know of much recent studies there yet, but I know that neurosurgeons are interested in this region to see what happens when you cut it out or spare it. So, but I don't have any conclusive evidence. It's interesting. I think that's still to come.
I think we have to go for a final question. Kevin asked if about 50% of the participants that you presented in the TMS fMRI examination, the signal propagates to the subgenual anterior cingulate. And the question is whether that isn't the factor that determines the success in RTMS treatment of the left DLPFC.
That's the $10 million question here. That's indeed what we hoped. So I've done a study, it's being written up now in Hong Kong with 70 patients where we look at resting state connectivity between these two targets. And indeed, it seems to be the case that it can predict treatment outcome. Paul Fitzgerald recently wrote a paper on a similar approach that indeed the connection strength between these regions can be used to some extent predict treatment outcome. Yeah, but this research is still ongoing, but this is a very relevant question. Yeah, we'll hear more about that in the coming years, I'm sure.
All right, thank you, Bas. I think we have to move
that this is far not so factor. So there are 10,000 different factors which will...
Obviously, yeah, it's a few percent that you explain. Yeah, very good point, Nikolai. It's a factor, but one out of many. Yeah, you're very correct.
All right, I think we have to wrap up the questions for now.
Okay, then Jort, I think you and me should switch roles.
Sounds good.
So, Jort is going to give the next lecture and share his screen. Jort is a PhD student at the moment at UMC Utrecht, where he is working on clinical trials to see how our TMS can augment the rehabilitation of patients after stroke. Jort is not only a brilliant PhD student, but we were also able to recruit him part-time at Brain Science Tools BV, where he fulfills many roles. One is customer support, but also demonstrations, as you'll see later. So Jort will present you on the findings of his trial and also how you can use diagnostic navigated MRI in a clinical setting. Jort, the floor is yours.
Thank you very much for those nice words, Bas. Yes, I'll be talking also on behalf of Rick Dijkhuizen, who is the PI on this whole project. I'll give a short introduction and then also tell a little bit more about the group that I'm a part of as part of the University Medical Center and the Rehabilitation Center that we do most of the practical work at. But before I do so, I would like to present to you what our clinical goals are of the overlapping project. And in addition to that, I think this talk will be a good example of how you can use certain techniques that have been previously described by Nikolai and Bas. For example, the TMS motor mapping, but also the RTMS treatment paradigms like continuous theta burst stimulation. But first, yes, why do we do this? So I work mainly with stroke patients, and a stroke basically is a sudden loss of oxygenation of the brain tissue. And this can be either due to blockage of an artery, which we call an ischemic stroke, or due to bleeding, rupture of an artery, in case we talk about hemorrhagic stroke. And stroke is a leading cause of disability worldwide and one of the main factors for mortality in the Western world. And what we see in about 80% of all the stroke patients that are admitted to our center is upper motor impairment or general motor impairment. And this is severely affecting activities of daily living, but also subjective well-being, and also health-related quality of life. And also for patients themselves, they stress recovery of upper limb function as a priority in their rehabilitation program. So what we are specifically interested in is a loss of strength, or in other words, a hemiparesis of the body, loss of strength in one side of the body. And the main problem that we encounter is that even though there has been a lot of improvement in acute therapy, we see that after 6 months, still 75% of patients are left with some impairment of the upper limb. So that's quite a substantial group. And this really stresses the need for improved rehabilitation of upper limb function for these patients. So we designed a sort of research line from preclinical to clinical care or clinical research. So some members of our group look at preclinical research in animal models. We have some more fundamental work, which is part of our INOS trial. What I will be talking about most is the B-STARS trial, which is the randomized clinical trial that we're currently doing. And then we're also working at implementation of TMS treatment in a clinical setting. So really the end stage of this whole research project. And I'm mostly involved with the clinical studies, but also with the implementation. So before we move on, I would like to give a short introduction about the neurophysiology of stroke. So patients who present with loss of strength from one side of the body due to stroke, they often show an imbalance in activity between the two hemispheres. So what we often see is that there is decreased activity in the affected hemisphere and increased activity in the healthy hemisphere. And this has been described in several papers, and different modalities have been used, like PET imaging, fMRI, TMS, to visualize this imbalance. And an important factor in the modulation of this imbalance is increased interhemispheric inhibition. So if there is a decreased activity in the affected hemisphere due to the stroke, the inhibitory and the transcallosal inhibitory pathways cause an increased activity in the healthy hemisphere and vice versa. Yeah, and the imbalance of this activity, so this disruptor of the network activity, it's associated with poor recovery of motor function. So what if we were able to normalize this imbalance and restore this network activity? Can we facilitate motor recovery? That's one of the most important questions within this field. And to go a little bit deeper into the neurophysiology, we first have to look at what movement initiation looks like in a healthy individual. So what we see in healthy individuals is that there is a constant interhemispheric, a certain level of interhemispheric inhibition between the two hemispheres when a patient is in rest. But when a person, a healthy individual, starts moving one hand, for example, the left hand, activity is generated in the right hemisphere, and the interhemispheric inhibition from the left to the right hemisphere diminishes. And that's what I'm trying to show here in this graph. So whenever there is movement initiation, we see a reduction in interhemispheric inhibition from the contralateral hemisphere. So, but what is the problem in patients? What we see in patients when they try to initiate movement with the affected hand, we see that the interhemispheric inhibition from the contralateral hemisphere does not diminish and actually persists. And previous research has showed that the persistence of the interhemispheric inhibition correlates with the extent of the hemiparesis. So patients who show a persistence in interhemispheric inhibition upon movement onset with the affected hand, they show a poor recovery. So this whole imbalance in network activity opens a door for neuromodulation, and this has also been mentioned by Nikolai in his talk. So what we can do is either perform excitatory TMS of the affected hemisphere to increase the activity there through long-term potentiation mechanisms and eventually normalize the network activity. But what we can also do is perform inhibitory TMS on increased activity in the healthy hemisphere. One factor that has been shown to be really important in neuromodulation and stroke is the window of enhanced plasticity. So what we see in the time course after stroke is a period early after stroke, the acute phase, where there is cell death, and after that there is a window of enhanced plasticity which lasts around 2 to 3 months after stroke. So this window of enhanced plasticity it's characterized by a number of more neurobiological processes, which I will not go into depth about. But this window is certainly something that we should use to our advantage. And when we look at the different meta-analyses of rTMS treatment in stroke, we see that when we subdivide all the papers that have been published into application of the TMS treatment in either the acute, subacute, or chronic phase, we see that the effect size is higher when TMS treatment is applied in this acute phase compared to when it's applied in the chronic phase. So this also shows that when you perform neuromodulatory treatment, you should do it in the window of enhanced plasticity. So you should do it in the acute phase. And then there's the question, what type of rTMS should we use? Either excitatory or inhibitory TMS. So there has been a new publication from Le Faucheur earlier this year, who updated the guidelines on therapeutic use of rTMS. And they concluded that contralesional inhibitory TMS in the post-acute phase was updated to level A evidence So this puts it in the same category as the left DLP-FC stimulation in depression. So this is really promising news for our field. And this also gives us reason to use an inhibitory TMS over excitatory TMS. This is not something that we knew when we started this trial. Other factors that we considered when deciding to pick either inhibitory or excitatory TMS is that when you decide to do excitatory TMS of the affected hemisphere, it's hard to determine your stimulation target unless you have MRI-guided neuronavigation early on. And that's not always the case. Often patients have very busy rehabilitation schedules, so they're not able to get an MRI scan in a different hospital because there is no MRI scanner at the rehabilitation center. And another factor is that there are no complications in TMS dosimetry. A lot of patients, they don't show an MEP in the early phase and the acute phase, so it's hard to determine what TMS intensity you should use. That doesn't mean that we did not use neuronavigation. We performed neuronavigation, but we did it on a generic template, and then we at the later stage, when we did acquire an MRI scan, we registered the captured positions to the individual MRI. And the targeting was based on the location of the functional motor area, so where we found the maximum MEP. So we decided to go for inhibitory TMS, and like Nikolai mentioned, there are two possibilities here. There's low frequency TMS, which generally lasts 20 minutes, which is quite a long protocol for patients. So we decided to go for continuous theta burst stimulation because it's only 40 seconds. But if you look at the literature, there is a very similar effect on excitability of the stimulated cortex. So the protocol, in short, we used inhibitory CTBS treatment of the unaffected motor cortex, delivered within 3 weeks after stroke onset. And the CTBS treatment was combined with regular hand therapy that was part of their rehabilitative programs. So 2 weeks of 5 sessions per week. And we followed these patients up to a year after stroke. So what is our hypothesis? So we believe, so normally patients show a recovery curve, and over time the motor function increases. But what we hope to achieve with the treatment and exercise is that patients recover faster and recover to a larger extent. So we're trying to modulate the course of their recovery. This is the protocol. I don't want to go into details on the protocol here. What I would like to mention is that we did motor mapping at baseline. So we mapped the motor eloquent area in the unaffected and the affected hemisphere. And at the same time, this provides us information on cortical excitability of these patients. We of course did sensory motor function tests like the ARET, which is more common in Europe, but also the Fugl-Meyer, which is a very popular tool in the US. And we performed MRI, which was optional for different time points to measure lesion size, to perform navigation, to determine white matter integrity, etc. So we have a randomized controlled trial. At baseline, patients are divided into an active treatment group and a sham treatment group, and our primary endpoint is at 3 months. So at 3 months, after this window of enhanced plasticity, we determine whether the active treatment group has recovered more than the sham treatment group. So unfortunately, I won't be able to share data that is directly related to the treatment groups because we haven't finished our inclusions yet, but I will be able to show you some data related to general recovery of these patients. So what I already mentioned when I was talking about our protocol is that we are doing TMS motor mapping before the treatment and after the treatment, up until a year after stroke. And why do we do this? Because Bas already mentioned, you can map changes, sort of cortical reorganization of the motor area over time. A very interesting paper that is very visual in the way that they describe this cortical reorganization is this paper on cortical reorganization in non-human primates. So what they did is they did invasive stimulation of the precentral gyrus or the motor area in these non-human primates, and they mapped different muscle groups. So for example, this dark brown area is the representation of the digits, while this light blue area is more as representation of more proximal muscle groups. And they induced a stroke in this area right here. After that, they did rehabilitative therapy for these non-human primates. And they again mapped the representation of these different muscle groups. And you see that, for example, the digits have expanded into more proximal representations. And other than that, you see that the infarcted area does not represent function anymore. So this cortical reorganization is clearly visible here, and this bar graph nicely shows their findings. So, of course, invasive stimulation is impossible to do in patients, but with the transcranial magnetic stimulation method that Bas described using navigated TMS, it is possible to map the motor area. So how does that work? You have, like already described, stimulation of the motor cortex, you get an MEP. If you combine that with MRI-guided neuronavigation, you can generate a map of cortical targets that you stimulated, and each of these flags represents a single stimulus. And the color of these flags represent the amplitude of the response. So if we filter out all the responses that do not exceed a certain threshold, we see that the maps, the MEPs that were made are nicely located within the precentral gyrus of this certain individual, a healthy individual. So if we do that in patients and we do it at baseline, but also at other time points after stroke, we can map the cortical reorganization of the functional motor area. And we can relate this to spontaneous recovery, but eventually also to whether these patients receive treatment or sham. So this is not the first time that something has been done like this. There are already papers out there which have correlated changes in the ipsilesional motor area to improved hand function. But most of these papers only measure changes outside the window of enhanced plasticity, so in the chronic phase, generally small sample sizes. So the evidence is not very convincing yet. But we have also mapped this, and what I'm showing you here is the displacement of the center of gravity of the motor area in a certain anatomical direction, because we believe that this shift is restricted based on anatomy. And what we see in the unaffected hemisphere is that there is displacement, average displacement of 3 millimeters. And this is something that we generally see in healthy individuals when measured over a period of a year. So when we look at the affected hemisphere, we see that early on there is this same shift, but this increases over time. So when you look at the X label, this goes from 3 weeks, so within the first 3 weeks, to up until a year after stroke. So we see that this displacement from the baseline motor area increases over time. So this is an interesting finding, and this is only preliminary because we haven't finished all the inclusions yet, but this is reason for us to look into this in more detail. But I'm going to leave it at this. And yeah, especially with regards to whether patients receive the CTBS treatment or not, we are definitely really interested to see if there is a more pronounced cortical reorganization in patients who did receive CTBS. I'm not sure how much time I've left. Yeah, there was a question during an earlier talk about what are barriers in applying TMS in stroke patients. So we are also looking at implementation into the clinic because we would really like to see TMS treatment getting picked up in more standard care. So what we're currently working on is investigation among healthcare professionals to identify what are barriers and facilitators in the implementation of TMS treatment. And we already finished qualitative research among patients to identify what their expectations are and their experiences are with TMS treatment after stroke. So what we got from patients, and among the 13 patients participated, they described the treatment as comfortable and painless. They sort of experienced it as a meditative moment because their rehabilitation schedule is normally very busy. And there was providing additional information on how it works definitely helped reduce anxiety for the procedures. So yeah, that's my talk for now. Are there any questions? You're muted, boss.
Thank you, Jort. Excellent overview of a really nice trial. There is a question right now. Someone posts lots of pluses in the chat window. Okay, so investigators tried down-regulating the less affected hemisphere in the niche trial. There are improvements in both groups, sham stimulation and contralesional inhibitory TMS was the same, Harvey et al. 2018. This goes against the inhibitory TMS hypothesis. Have you read about this trial?
I'm not specifically aware of this trial per se. What I can say is that when you look at the more recent meta-analyses in combination with the clinical guidelines, I definitely think there is evidence for inhibitory TMS of the contralesional hemisphere. It's hard to look at individual trials, and I think we should try to combine different findings into meta-analyses. And the literature guidelines are definitely really promising in that regard.
All right. Thank you. I believe, let me see, there are no other questions at the moment. I might have one myself.
Let me comment last question.
Sure.
So the sham stimulation is tricky itself, and there is many discussion about what should be sham, because when we say sham stimulation, we just say sham, but what is reality behind? And there are many discussions about that, and looks like in some such trials, like niche trial, the sham stimulation was too intensive so it has an effect itself, even though it's called sham.
Thanks, Nikolai. Yeah, there are two other questions. Yeah, we can answer one of them, I think.
You want to go back to what Nikolai just mentioned?
No, yeah, I see that somebody else asked a question. One question is, how do you deliver TMS in stroke patients who do not show MEPs? Yeah, that was our main consideration, to stimulate the contralesional hemisphere. So in the contralesional hemisphere, there is always the possibility to evoke an MEP, and therefore it's always easy to dose the TMS. In the affected hemisphere, I'm not sure if Nikolai can comment on it, but yeah, if there isn't any MEP, it's not possible to determine the dose.
All right, I have one myself. Oh, sorry, if you came in, then I'll go for a question asking, do you believe TMS research in stroke can shed light on TMS for depression?
It's a very good question. I think when we learn from stimulating a network, and I believe that if it's the depression network or the motor network, that the ways that the brain responds to the TMS signal is similar, and that you can definitely extrapolate findings to different networks. But yeah, you should always be very careful in that regard.
Okay. And finally, there is a remark for you that Cambridge et al. Did research on the contralesional hemisphere. More impaired participants do not benefit from downregulation of contralesional cortex. Just for your information.
Yeah, I can comment this from pure clinical point of view. So if you let's say you have a clinic which starts doing stroke patients, and of course you would try to avoid affected hemisphere. And the very first thing which you would do in real life is the very first thing you would try to play with the non-affected hemisphere, right? And this is why most publications and most work is done on this side. But of course, we see a lot of nice trials, nice publications about results in affected hemisphere. And I personally believe that we should have better results when we do that. So people just afraid to touch it. This is my point of view.
Okay. Then, yeah, we're only a few minutes over time. Thank you for everybody to keep to the tight schedule we have here. And also, again, once more to Jort for his really nice talk on the BSTARS trial. I want to add here that Professor Rick Dijkhuizen, who really is the mastermind behind this trial, was supposed to give this talk. So Jort took over, so he's really leading this stroke research. So he couldn't be with us, unfortunately, due to circumstances, mainly childcare, I believe. Having said that, if there are no further questions, we will now go to the live workshop, where we will actually demonstrate you several of the techniques that we've just been talking about. With this, I'd like to give the floor back to Jort, assisted by Sjors Hooiberger. Sjors will show us the equipment. So please bring on the demo room screen, Alexei. There you go. And Jort will comment on what Sjors is doing and showing you. So Sjors is also a master student in the BSTARS trial and also working at our company as a junior sales associate. Jort and Sjors, the demo room is all yours.
Thank you, Bas. I would like to ask everyone to go into full screen mode, and then when Sjors will be sharing his screen, you can have the video feed. You can have it also present on your screen, and you can also increase the size of it. Hopefully that works for everyone, but otherwise the video feed is relatively small when Sjors will be sharing the screen. Yeah, so right now, Sjos is standing near the chair, the treatment chair. Hi, Sjos, thanks for doing the demo. Behind the chair is a small transmitter box. Yes, that one. And this transmits an electromagnetic signal, which captures the positions of the sensors. Two of the sensors are positioned in the headband, and so they track position of the head and potential head movements during the navigation session. And the other one is attached to the TMS coil to capture the location and the movements of the TMS coil. So in order to align the patient's head with the MRI in the neuronavigation software, we use the pointer on the right, and whenever you capture a position on the face, you do it with a remote control. And our electronics are mounted to a trolley, so that's basically where all the information of the sensors is integrated and being fed to the PC. Then we have the EMG device. The EMG we currently cannot showcase, unfortunately, because we cannot be in the same room at the same time and keep a 1.5 meter distance as advised by our government when we demonstrate. But instead, we'll show a short video on how the TMS motor mapping works. But that doesn't mean that we cannot show the demo on our fake patient. But the EMG is connected to the PC as well, and it feeds information of the MEPs to the software. Then on the shelf, we see the TMS device. It's also integrated with the software. So whenever you discharge the device, it will capture the position of the TMS coil and capture the characteristics of the MEP.
Yes.
So now Sjos is sharing the screen. So now you would be able to open up an additional window, and you can kind of enlarge it so you can see the video feed and the screen at the same time. I think this only works in full screen mode. So this is what the software looks like. And Sjos will now load the data. So we support NIFTY data and DICOM data, and you can separately load the anatomical scan, which is basically the raw T1 weighted anatomical image. It will give a preview of what the data looks like and also assesses the quality. So if it's green, that means that the quality is sufficient for navigation. So now in the bottom, you can see that it's loading the T1 and weighted scan, and when it's finished loading, it will show a 3D surface reconstruction of the head. At the same time, it will calculate normalization parameters so that we can inverse normalize MNI coordinates to this individual patient. We're for now going to load a segmented brain that we acquired from an external package, but our software also allows brain segmentation within the software. If you're interested in that, you can always subscribe for an online demonstration for our website. So now we have a 3D reconstruction of the brain as well. So now it's time to either load a pre-configured set of markers based on the MNI atlas, or you can manually create them yourself, but we'll go for the pre-configured set at first. There are multiple options, but we'll go for the set with 6 markers now. 6 is generally advised. If you use more markers, the accuracy will slightly increase, but this kind of saturates around 6 markers. Because this is a pre-configured set of markers, it's good to always manually check whether the markers have been placed correctly and adjust in where needed. Those are already quite accurately placed. Placement of the markers is really a matter of personal preference. What's important here is that the markers that you place inside the MRI scan are positioned in a way that you can also easily identify them on your patient's head. So that's why the nose tip is a clear anatomical feature together with the nasion and aspects of the ear. But for example, the Indian, it's harder to identify on the patient's head. So the same goes for the target. So now we'll go for the left DLPSC. So we're interested in stimulating, performing a therapeutic session for a depressed patient. Shows repositioned it slightly. It's depending on your preference, but Fitzgerald guidelines prescribe that you position it 3 gyri anterior of the precentral gyrus and within the middle frontal gyrus. So that's where it's located now. And then we'll place 1 target at the primary motor cortex for the motor mapping procedure. And it's nicely positioned within the hand knob of the precentral gyrus. So we'll start the tracker. So this is simply initialization of the electronics. And whenever it's finished, it will ask us to capture the first marker as part of the set. So in the bottom left, you can see that the software asks us to capture the nasion, and that's also highlighted in the 3D image. So what SCHOSS will do is point with the pointer at the nasion, and then using the remote control, you can capture that position. And we hear an auditory confirmation that this has been done. So we'll ask you to capture all the different markers on the patient's head. And when this has been done successfully, it will also show you that the alignment was performed successfully. If you, for example, captured one of the markers in the wrong position, there's a possibility to go back and recapture a marker. But now everything was done successfully, and that means that the software will start to stream the position of the TMS coil automatically. So we already see it hovering over the patient's head. So what we see is a TMS coil model, but what we're most interested in is the yellow beam that is at the TMS coil isocenter. And this yellow beam has a small flag protruding from it, which indicates the direction of the induced current. What SCHOSS enabled is the distance to target feature. So we'll measure the distance between from the TMS coil isocenter to the target. So now there is a 4 millimeter distance between the coil and the target. And whenever SCHOSS moves away the TMS coil, you get an auditory warning that you should reposition the TMS coil. This feature also provides information on the angle and on the depth. All right. Yeah, so whenever the TMS coil is positioned accurately, you can start the TMS treatment. We will not go into the details of how to do that now. This is something that we will talk about in great detail during our course in December. And for stimulation for motor mapping, we have an interesting feature that Bas has already talked about in great detail. Shows us now navigating to the primary motor cortex, and whenever he discharges the coil, the software will automatically place a flag onto the cortical surface. And this flag will provide information on the MEP. So we don't have an MEP for now because our friend is not physiologically active, unfortunately. This will contain information on the TMS target location and on the orientation of the TMS coil. We're resetting the TMS device, it seems. I believe there are some connectivity problems, but it's hard to assess from here. Can you zoom in a little bit, Jos? So now we have stimulation targets positioned on the cortex whenever Jos discharges the coil. And they're all white because in this case, we don't have the actual EMG attached. If you go to the, and now we are only seeing the TMS coil isocenter. So it's this basically to enhance visibility of what you're doing. So in this way, you can systematically map out the motor cortex and get an idea of where the motor eloquent cortex is represented. And you will get a list of MEP characteristics for each of the TMS targets. So I think now would be a good time to switch to our video where we can actually show this in on a real person. Welcome to this instructional video on how to map the motor area using the Neural Navigator navigated MEP. We will start by preparing our patient by placing the EMG electrodes over the muscle of interest. In this case, we are mapping the first dorsal interosseous muscle of the right hand. We attach the ground electrode to the wrist. Next, we place the recording electrode in black over the muscle belly and the reference electrode in red over the tendon. Now that the EMG electrodes have been placed, we can start the NeuroMap software. This software allows us to record the EMG traces. Go to Magnetic Stimulation and select the first dorsal interosseous muscle from the list. Since we have already placed the electrodes, we can check the impedances by clicking on the Check Impedance icon. If the impedances are green, the electrodes are placed correctly. Now click the play icon to start monitoring the EMG trace. In case you have not yet set up neuronavigation, you can find instructions on how to do this in our video on how to perform neuronavigation. Now we can start navigating. Whenever we deliver a TMS pulse, a sphere will appear on the brain's surface. The position of the sphere indicates where the TMS pulse was delivered. The color represents the amplitude of the evoked response in the muscle. We can stimulate different targets in the brain to generate a delineation of the area in which responses are evoked in the muscle. For each stimulation target, you can find the machine output, the amplitude and latency of the evoked response, and the X, Y, and Z coordinates of the stimulation target in the table in the MEP tab. You can also view the waveform in the panel above. To explain additional features of the software, we will load a different dataset. In this dataset, a grid of points has been stimulated. You can adjust the color scaling of the spheres by changing the minimum and maximum amplitude of the evoked response. You can also select a different color map from the drop-down menu. You can adjust the visualization of the stimulation targets by changing the spheres to flags. The advantage of using flags is that they point in the direction of the TMS-induced current. If you are using multi-channel EMG, you can visualize different channels by selecting the channel number in the drop-down menu. Finally, you can filter the data based on features of the data. For example, you can visualize all the responses with an amplitude of more than 1 millivolt. Lastly, you can save and load the data by clicking on the Save and Load button respectively. Thank you for watching. Thank you very much, Jos. There is some very good news that we would like to share with you that we got last week, and that is that our system has FDA approval now. So that's very good news that we wanted to share with you as well. Maybe Bas wants to quickly mention something about that as CEO of the company.
Thank you, Jos. Well, at the moment, I'm CSO CTO, so as you know, as of a few weeks ago. Yeah, indeed, we're really happy that after 1.5 years of work together with Soterix Medical from New York City, that the FDA has now cleared the combination of the cloud TMS or NeuroSoft TMS system and this navigation system for clinical use, both in treatment of depression, but also for motor mapping. So yeah, that's the second system that has this clearance in the entire United States at the moment. So yeah, that's really good news for us. And we're happy we were able to show also a largely US-based audience that this is indeed now available to you through Soterix Medical. Having said that, I'd like to thank all the speakers and demonstrators of today for a session that I believe went really well. I hope it was of interest to our more than 90 participants. I also like to thank the Neuromodulation organizers for making their platform available online in these trying times. It's really a format, I think, that deserves continuation and copying. So thanks, Neuromodulation. And last but not least, thank you, participants, for spending these hours with us, probably also from a home quarantine situation as well. So let's do this more often. Approach us through our website, brainsciencetools.com. We're always available for additional questions. I really like the interactive nature of today's chat session, so lots been going on. These videos will be recorded and are supposed to be published on the Neuromodulation website soon. If there are no further questions, I would maybe give the word to Nikolai, who has a few things to add from Neurosoft. Nikolai?
I want to thank everybody who was attending today's session and everybody who was presenting today. And I really want to encourage all of you to visit our course in December, which will be in December 8 and 9, and you can visit tmscourse.nl for more information. And have a great day. Thank you very much.
Thank you, Nikolai. With this, the Neuromodulation session comes to an end, right on time. I wish you a nice evening. Stay safe, and good luck and a lot of strength in these trying times. Bye-bye. Take care, and so long.
Bye.